Hello there! Guess what? It's chapter 26! Yay! This chapter we're going to be talking about bacteria and archaea. So, by the end of this chapter, you should be able to explain how bacteria and archaea are the most important, diverse, and abundant organisms on the planet. You should be able to describe the 6 processes that bacteria and archaea use to produce ATP and obtain the carbon building blocks that they need. You should be able to describe the key roles that bacteria play in agriculture, in bioremediation, and in human health. Alright, so there are 3 domains of life, right? We learned about this before. 2 of the domains are bacteria and archaea, and then the third is eukarya. So today we're going to be learning about bacteria and archaea. Bacteria and archaea are prokaryotic. Of course, eukarya is eukaryotic, right? So today we're covering both of the prokaryote groups. Bacteria and archaea all lack a membrane-bound nucleus. So they have genetic material, but it is not contained within a membrane. And most bacteria and archaea are unicellular, meaning composed of only one cell. Alright, so there are other distinguishing features of bacteria and archaea, including molecules that make up the cell walls and plasma membranes, and also some of the machinery used to transcribe DNA and translate messenger RNA into proteins. So we'll talk more about this later, but there's particular DNA sequences in ribosomal RNA that we use to distinguish between these groups pretty easily. Okay, so why should we study bacteria and archaea? First of all, biological importance. They are extremely abundant. They are everywhere. They can also live in really diverse habitats, so you have some bacteria and archaea that are extremophiles. Archaea are especially good at this. This means they can live in places that are extremely harsh and nothing else can live. They have medical importance, so we're going to learn today about Koch's postulates and germ theory and pathogens. They can also be used in bioremediation, which I'm going to talk about. Okay, so the oldest fossils that seem to be of bacteria are about 3.5 billion years old. And so what we see here are these weird loafy looking things, right? And this isn't the bacteria itself. Instead, this is like kind of a layer of bacteria that is producing these kinds of rock as a byproduct of some of their metabolism. Okay, so that's our earliest evidence of a bacteria. And we don't see eukaryotes in the fossil record until 1.7 billion years later. Oh, I'm sorry, and these are some other early evidence of bacteria. All right, so currently there are 10,000 species named and described, but there are likely to be hundreds of thousands of species of bacteria. We tend not to give them names as often as we would eukaryotes, so, we just kind of call them different strains and don't bother and rename them all the time. So just to give you some sense of scale, if you looked inside of the human large intestine, you would find approximately 1,000 different microbes, meaning microscopic organisms, many of which are bacteria and archaea, and 700 microbes are normally found in the human mouth, including bacteria and archaea. Alright, so on Earth, bacteria and archaea are the dominant life forms. If you added up the total volume that all the bacteria and archaea took up on Earth, they would completely dwarf everything else. If you were to take a teaspoon of soil...
Is that what TSP stands for? I thought that was tablespoon.
No, tablespoon is TB something else. Make sure you get this right next time you cook.
Okay.
Okay, so there's billions of microbes per teaspoon of soil. If you were to get a single milliliter of seawater, you would find more than 10,000 individual marine archaea in it. The ocean bacteria and archaea are 10% of the world's total live mass. And we know that the bacteria and archaea can live almost anywhere. So as far as we've dug below the Earth's surface, we have found bacteria and archaea. We've even found bacteria and archaea preserved in Antarctic sea ice. There are entirely new phyla of bacteria and archaea that have been recently discovered. So discovering something that is so different from everything else that it gets its own phylum is very rare, but this pretty much only happens with bacteria and archaea. And the people who study bacteria and archaea are called microbiologists, so they study microbes. Okay, so some prokaryotes can live in really extreme environments, and we call these guys extremophiles. And some of the examples of extreme environments that have prokaryotes in them are hydrothermal vents, so these like super boiling hot vents underneath the ocean, in places where the pH is less than 1. So is that very acidic or is that very basic?
Oh, that's very acidic.
Yeah, that's acidic. We have found things that are under 0 degrees Celsius underneath the Antarctic ice. We have found extremophiles living in water that is 5 to 10 times saltier than seawater. So for example, we can see here these salt ponds here are pink because there are archaeal cells that are coloring these crystals of salt. So that's why these are pink. So clearly archaea are living their best possible life in these pools of salt. We think that that prokaryotes, especially extremophiles, may explain how life on Earth began. And so when we go and look for extraterrestrial life on other planets, we're always looking for prokaryotes because we think that that was the first life on our planet. We're also going to talk a little bit about some of the extremophile enzymes that extremophile prokaryotes produce that we have been able to kind of hack into to be able to use for our own industrial chemical processes. All right, so I probably don't need to convince you that bacteria can cause illness in humans. So this is just some examples. So for example, Clostridium, there's a Clostridium bacteria that causes tetanus, spirochetes that cause Lyme disease and syphilis, pneumonia is caused by bacteria, as is tuberculosis, leprosy, acne, gonorrhea, the plague, okay? So hopefully I have convinced you. Don't memorize this table or anything, but I just put this up here to make sure you understand that we care about bacteria for many reasons, one of which is that they are of large medical importance. Okay, so I'm going to introduce you to the idea of Koch's postulates now. And so Koch was a researcher who wanted to be able to figure out how you can definitively say, like, okay, I see a bacteria, I see a sick person, how do I know this bacteria caused the sickness in this person, okay? So he came up with his postulates to determine the causative link between a specific disease and a specific microbe. So the first step that you have to demonstrate is that the microbe is present in individuals suffering from the disease and absent from healthy individuals, right? So you have to be able to find the bacteria, or it could even be a virus or archaea, in people who are sick, but don't find it in people who are healthy. All right, next, you have to be able to take this microbe out of the sick individuals and isolate it and grow it in pure culture, meaning grow it in a petri dish, on like a solid agar petri dish or liquid broth, and be able to grow it up. And then you take organisms from this pure culture, and by the way, what I mean by pure culture is you grow this microbe in a petri dish, and then you kind of isolate just that one colony from the other ones, so that you have a tube that is only that one very specific microbe that you're interested in. Okay. And so then if you take organisms from pure culture, from that petri dish, and inject them into a healthy experimental animal, that animal should have the symptoms of the disease, okay? And then finally, once that experimental animal has the disease, you can take a sample from that animal, and you should be able to find that microbe again in that animal. So that microbe went into the animal, it made the animal sick, and then it reproduced, so that when you take a sample from the animal, you'll find that microbe again, okay? And you also have to demonstrate the microbe that you got out of the experimental animal is the same as the microbe that you got in step 1. Okay, so those are Koch's postulates. So you have to be able to demonstrate those 4 things in order to say that a particular microbe caused a particular disease. And so this might seem really picky, but I should point out that frequently there are many different kinds of microbes that can affect somebody who's already sick with something else, right? And so it's really important to figure out if the person got sick and then their immune system was out of whack and that made them susceptible to this other microbe, or maybe a microbe that already was in their body, was able to take over because they were already immunocompromised from a different illness, or if the microbe caused the illness, okay? Okay, this is a lot of words, but let me show you what's going on. These are Koch's postulates at the top, so you can see them, okay? So that's what we just did. All right, so then I have a series of questions here. The yellow questions go with the first postulate, and then this pink or whatever question here goes with these postulates 2 and 3, and then the green ones go with postulate 4, okay? All right, so let's think about the first question. Well, let's think about the first postulate and the first question. So the first postulate is that a microbe present in individuals suffering from the disease, you must find the microbe present in individuals that are suffering from the disease, and you don't find the microbe in healthy individuals. All right, so I'm going to ask you guys, if you find the microbe in someone who doesn't have the disease, what does this mean?
Maybe they're just not sick yet, but they're gonna get sick.
Okay, it could be. It could be that maybe the person without the disease is going to get the disease later. Or what else could it mean? It could mean that we just all have that microbe in us. Like whether you're sick or healthy, everyone has it. Yeah, that's true. We have plenty of microbes that live in our body all the time, right? Anything else? Okay, well, I think that's good enough for that one. All right, next. If you don't find the microbe in a person with the disease, what does this mean?
Wait, you don't find the microbe in the person with the disease?
Yeah, so what does that mean?
Maybe it means that the microbe didn't cause the disease.
It could mean the microbe didn't cause the disease. What else?
I don't know. It could mean that you don't need very much microbe to get the disease, and there's so little you can't even really see it.
I mean, I guess. That doesn't happen very often, but it's possible.
Yeah.
Okay. So which of those two things, finding the microbe in someone without the disease or not finding the microbe in a person with the disease, which of these is stronger evidence that the microbe did not cause the disease? Oh, look, it says case. Did not cause, oh here, I'll use my pen. Did not cause the disease.
Well, it seems more likely that someone could have the microbe and then not get sick. Maybe they have immunity to it, or maybe their body is fighting it. So it seems like the other one is stronger. If you get a disease but you don't get the microbe, that makes it seem like probably that microbe didn't cause the disease.
Yeah, I agree with you. I think the second one is better evidence that the microbe did not cause the disease. All right, so let's look at the second and third postulate. So the microbe has to be isolated and grown in pure culture away from the host, and then you inject the microbe from the pure culture into a healthy experimental animal, and the animal gets a disease.
That's terrible. You shouldn't do that.
Well, he's helping science.
Okay, if he's helping science.
All right, so my question is, why do you have to culture the microbe in a petri dish? Why not just take a blood sample or something, or a swab, from the sick person to an uninfected person? Oh, I know. It's 'cause, then you won't know which thing that's on the swab or the blood sample caused it. So, you could transfer the thing that you think is the microbe and then a bunch of other stuff, and it could have been one of the other things that caused the disease. Yeah, that's true. All right, so then for the last postulate, you isolate the organism, the bacteria or archaea or whatever, from the diseased experimental animal, and you grow it up again in pure culture, and then compare it to the original organism and see if it's the same. All right, so why do you need to culture the disease from an experimentally infected person?
'Cause you gotta find out why they're sick.
Yeah, you gotta find out why they're sick, right? All right. And so what does it mean if you can't find the microbe in the experimentally infected person?
Oh, that's very suspicious. That could mean that the reason why they're sick isn't due to the microbe.
Yeah. That could mean that the reason that they're sick isn't due to the microbe, but due to something else, right?
Yeah.
Okay, so now that we've talked it through, I hope you guys understand the logic behind Koch's postulates. Let's talk a little bit about the germ theory. So Koch's experimental results were the first test of what we call the germ theory of disease. And it is, remember we talked about pattern and process, right? So the pattern component, the thing that people observed, was that some diseases are infectious, okay? The process component that explains why some diseases are infectious is that you have certain bacteria and viruses that are transmitted from person to person and then grow within those people, okay? And so we know that infectious diseases tend to spread in three main ways. They're either passed from person to person, like by breathing infectious droplets on them, for example. They can be transmitted by bites from insects or animals. Can you think of any examples of that?
Rabies!
Yes, rabies is a disease, yes. What else? What do mosquitoes potentially give us?
Lyme disease!
No, they don't give you Lyme disease. Ticks can give you Lyme disease. What can you get from mosquitoes?
Oh, malaria!
Yeah, malaria. Or Zika, right? And you can also acquire infectious diseases by ingesting contaminated food or water or exposure to microbes in surrounding environment, okay? Okay, so there are plenty of bacteria that live with us all the time and don't give us any problems, right? But some bacterial cells are pathogenic. So if something's a pathogen, it means that it makes you sick, right? So, we refer to bacterial cells that are pathogens as having virulence, meaning the ability to cause disease. And virulence is a heritable trait that varies from individual to individual. So the virulence might be, for example, having a particular kind of a chemical in the coating on the outside of the bacteria. And the bacteria divides and passes, divides and makes babies like by division, and then passes on that virulence gene, which is expressed in the offspring, etc., right? It's variable, meaning some individuals may have that gene for that coating that makes them, harder for the host to kill, and so they're variable. So sometimes you find species that have both pathogenic virulent strains and harmless strains. So a good example of that is E. coli. And, every once in a while you'll hear a story in which people will eat a hamburger that has been infected with E. coli, and then they get really sick, and then people get really worried about E. coli, right? But it should be noted that not all strains of E. coli are bad. We have strains of E. coli that live in our gut all the time, and we totally need them for helping digest food. Okay? So within E. coli, when people have sequenced the genomes of pathogenic strains, those genomes are larger than the genomes of the non-pathogenic strains because the pathogenic ones have acquired virulence genes. So we can actually see their genomes are bigger because they're genome plus virulence gene. And an example of a virulence gene would be a gene that maybe codes for a protein toxin. So the bacteria are just there living their lives, but they produce some kind of a protein toxin that makes us sick. All right, so bacteria have this really cool thing called endospores. So these are these tough, thick-walled dormant structures formed during times of environmental stress. So this bit in the middle here is the endospore. And so it's kind of like having a little tiny locked room within your house where you can go, I don't know, to hide or something like that. And so locked within this room are all of the bacteria's goodies, including the cell's DNA, its RNA, its ribosomes, and its enzymes, okay? And so when, times get really tough and the bacteria starts to form an endospore, the metabolic activity stops, meaning it stops eating food and making ATP, and then the outside of the original cell will break down. When it starts to form this endospore, the endospore and everything inside of it is resistant to high temperatures, UV radiation, and antibiotics. And then when conditions get better, then the cell can start to go back to actively dividing and live its life like it was before. And so the fact that bacteria can form endospores is really important in transmitting disease to humans, right? So you can, for example, freeze these or dry, if you're bacteria, you can freeze out or dry out and then rehydrate or, kind of thaw and then resume your kind of activity later. So this makes bacteria really hard to kill. All right, so antibiotics are molecules that kill bacteria or stop them from growing. And so these are produced naturally by some soil bacteria and fungi. So this is a kind of a fungus that happens to produce an antibiotic that we like. And so the first antibiotics were discovered in 1928. And by the 1940s, we were using them regularly to help with disease. So their extensive use in the late 20th century, in not only like clinics to help human health, but also to be fed to animals to keep animals healthy, has led to the evolution of drug-resistant strains of pathogenic bacteria. So if you go back to chapter 22, we give some examples of this. I should also point out, the reason why we tend to give animals like cows antibiotics isn't necessarily because they're sick. We have found that if you give cattle antibiotics, they actually grow faster. So in case you're curious why we're giving antibiotics to animals in large amounts, this is why. All right, you can sometimes find bacteria involved in biofilms. So biofilms are bacterial colonies that are meshed into polysaccharide-rich matrix that shields the bacteria from antibiotics. So this matrix may be made out of not just bacteria, but it could be made out of fungi. In this case, maybe there's some algae involved in it and other kinds of protists. And so you get this like community made out of bacteria and other stuff that coats things like the hulls of boats and the inside of pipes. So I don't know if you've ever opened up a pipe and found something sticky and gloppy, but that's probably what you're looking at. All right, so bioremediation refers to using bacteria and archaea to clean up sites polluted with organic solvents. So there are a lot of water pollutants that would kill a eukaryote like us and don't dissolve readily in water. And so these will tend to accumulate in sediments, right? So they never really go away unless you do something about them. However, there do exist populations of bacteria and archaea that can kind of digest and metabolize the materials that are in spills. And when they, kind of digest the chemicals in the spills, the byproduct that the bacteria and archaea produce is non-toxic. So they can basically degrade the toxins and make them non-toxic. And so bioremediation can use one of two different strategies, or it can use both. The first one is that maybe you have a site that probably already has some naturally occurring bacteria and archaea there that could break down the toxin, but maybe the bacteria and archaea, they need a little food or something. And so you would go and fertilize the contaminated area to encourage growth of the existing bacteria and archaea, and then the bacteria and archaea would digest the toxin. Alternatively, sometimes people will actually spray or add bacteria and archaea that they know can deal with, certain kinds of contaminants and use them to break down things. And frequently the way that the bacteria and archaea are applied is through the form of a biofilm. So that's another bonus. You can make a biofilm of helpful bacteria or archaea and use that to decontaminate a site. All right, so there are multiple different approaches to being able to study bacteria and archaea. The first is called enrichment cultures, second, metagenomics, third, there's a kind of a human biome approach, and then the fourth is the use of molecular phylogenetics. All right, so enrichment cultures are very, very commonly used. So almost all described species that we know about of bacteria and archaea were discovered in controlled conditions in a laboratory. So nobody went outside and just pointed to the ground and said, I claim you, I have found a new discovery, right? We always have to bring these things into the lab to be able to see them in detail. So the idea of an enrichment culture, so a culture is anything you're using to grow any kind of living thing in the lab, right? And an enrichment culture means that you're going to set up your test tube with very specific living conditions, and you're going to figure out what can live there. And that will help you isolate populations of cells that can grow under those specific conditions. So these kind of enrichment cultures were what allowed us to discover thermophiles or heat lovers. So if you set up an enrichment culture that is at a very high temperature, then you can see what can live there, and that'll help you discover kinds of bacteria that live in hot places. All right, so this research question is just showing how you can use enrichment culture to discover new things. So the question is, can bacteria live a mile below the Earth's surface? Okay, so the hypothesis of the researchers was that the bacteria are able to use cellular respiration way below the Earth's surface by using hydrogen as the electron donor, and because there was no oxygen way down there, they used iron as an electron acceptor. Okay. So the null hypothesis is that bacteria from this environment are not capable of using the hydrogen as a donor and the iron as an electron acceptor. So the setup is to prepare the enrichment culture and give it the kind of things that we think that it needs in order to be able to reproduce. So we provide iron and hydrogen, and then raise the temperature above 45 degrees C, which is what we think the Earth is like, way that far down below the Earth. And then we add rock and fluid samples that were extracted from drilling operations. So people are digging 1,000 meters below the surface of the Earth and pulling up rocks, and then we kind of drop them in these tubes. The prediction is that black magnetic grains of magnetite will accumulate because the iron is going to get reduced by growing cells and then shed as a waste product, and so then you'll see the iron in the magnetite. And because the magnetite grows visible crystals, you should be able to actually see these cells, okay? And so the null hypothesis predicts that you won't see any magnetite and you're not going to see any cellular growth, okay? Okay, the results were, yeah, they saw cellular growth. They also found magnetite. And so this, in order for the magnetite to have appeared, something must have reduced the iron to make the magnetite, right? And so they conclude that at least one bacterial species can live deep below the Earth's surface because it was able to grow in this enrichment culture. And so, like, maybe if you took other samples or slightly different enrichment cultures, you may find other species. Okay, the second approach to finding bacteria and archaea is through metagenomics. And another name for this is environmental sequencing. And so we do this a lot. Basically what you do is you take a sample from somewhere and you don't know what's in the sample, right? So it's not like you're taking a blood sample. You're like just taking a scoop of soil or an ice core, okay? And so then what you do is you extract all of the DNA from that sample and get out all the inorganic material and other cellular material, only leaving the DNA. Chop the DNA up into little fragments, and then sequence them. And by sequence, I mean figure out what the string of nucleotides are for each of those fragments. So like T, C, C, G, A, T, etc. Okay, because we have lots and lots of organisms that have already had their DNA sequenced, and that information is widely shared. If you go on GenBank, you can see all of the sequences, everyone's uploaded. You can go figure out where your little pieces of DNA match up with things that are already in the database. And then that will tell you which particular organisms you have found in a place, or if you found something new that doesn't match anything else. All right, a third approach is called a microbiome approach. So microbiome is a community of microbes that are naturally found in the body or in parts of the body. And so I bet that the Human Microbiome Project must be pretty close to complete by now. And so they were trying to sequence all of the different kinds of things that they found in 250 healthy people. So this is an artist went and illustrated their ideas about the Human Microbiome Project. And so then they took samples from lots of different parts of the body, and they discovered different things about the microbiome in the different parts of the body. So, for example, in the human gut microbiome, there were 100 trillion bacteria and archaea found. They found some microbes that have the capacity to make us sick, but we have lots and lots of other microbes that we need in order to stay healthy. They also found kind of some interesting relationships to other problems. So they found that changes in the gut microbiome have been linked to both inflammatory bowel disease and obesity. So people who tend to have for example, inflammatory bowel disease, have changes in their gut microbiome compared to other people, and similarly with obesity. Okay, next, there are 3 domains of life, right? Bacteria, archaea, and eukarya. And so we need to talk about what the relationship is between these 3 domains. So one of the oldest hypotheses was that there were 2 domains, just the prokaryotes and the eukaryotes. Okay, so that's the oldest one. A slightly more recent hypothesis is that there were 2 domains, but the 2 domains consisted of bacteria being the one of them and everything else being the other one. All right, a third hypothesis is that archaea is the root of the tree and that bacteria and eukarya are more closely related to each other than either one is to archaea. And then finally, the 3-domain hypothesis, which is the one that we tend to kind of go with in more recent times, and our book supports this one as well. So Carl Woese had a data set in which he sequenced the small subunit of ribosomes. So, do you remember ribosomes? What does a ribosome do?
It translates.
Yeah, it translates what?
Translates messenger RNA into amino acids.
Yeah. So you remember it looks like a little snowman?
So cute.
Yeah, it's a little snowman. And then there's a little piece on top and a big piece on the bottom that snap together, right? The little piece on the top is called the small subunit. And there's little sequences in there. So there's 16S and 18S RNA. And so we can take these sequences and compare them across many, many species, including bacteria, many species that are within the 3 domains of bacteria, archaea, and eukarya. And then the take-home message is what we have found is that actually archaea and eukarya are more closely related to each other than either one is to bacteria.
That's crazy!
Why?
Because bacteria and archaea look alike. They're both unicellular. They're both prokaryotes.
I know, it's crazy, but the information tells us what it tells us. It tells us that archaea and eukarya are more closely related to each other than either is to bacteria.
Okay, fine.
Okay. So let's talk a little bit about diversity in bacteria and archaea. So there is ecological diversity, which can have global impacts. So bacteria and archaea make all sorts of really awesome enzymes that allow them to live in like crazy places, right? So, for example, the archaea that live in the salt flats or, in the cold weather, they have enzymes that allow them to be able to survive in those places, right? The ones that we use for bioremediation are able to take toxic compounds that kill everything else and make them into food. And so the fact that they have this complex chemistry and that there's so many different kinds of bacteria and archaea make them something that we can use for global change. All right, so the second thing is that they're very diverse because they can get genetic variation through gene transfer. So bacteria can, I'll show you in a minute, ways that bacteria can get genes from other bacteria. They also differ morphologically, so I'll show you some of the ways that they look differently. They differ in metabolic diversity, like what they use to be able to get their ATP from and also their carbon-carbon bonds. Okay. First, why is it important for organisms to be genetically diverse? So they can survive some kind of environmental problem. Yeah. So how does being diverse help them survive an environmental problem?
Well, maybe, one of them, if there's a lot of them and they're different, it'll have a mutation that'll allow it to survive.
Yeah. So what is the ultimate source of all this genetic variation?
I just said it, it's a mutation.
Yeah. Okay, so how does meiosis increase genetic variation in eukaryotes?
It doesn't give you it doesn't give you new mutations, but it makes it so you can make new combinations of the variations you already have.
Yeah, so you can have offspring that are different combinations of different kinds of genes. All right, so for prokaryotes that reproduce by binary fission, how does reproduction affect genetic variation?
Wait, what?
Like, when a prokaryote like a bacteria reproduces.
What's binary fission?
Well, what's binary mean?
It means two.
Yeah, what's fission?
It means to split.
Yeah, it means to split, right? So prokaryotes reproduce by splitting in half, okay? So how does reproduction affect genetic variation?
It doesn't affect it at all. They're just the same. They have the same genes as their parents.
Mm-hmm. Right. All right, so bacteria do not get new genetic information in their offspring through reproduction. They have to do something else, right? They get genetic variation through gene transfer. So there's 3 ways they can do that. The first one is called transformation, and this is when bacteria or archaea take up DNA from the environment that has been released by cell lysis or secreted. So you can think about the bacteria and archaea as being like little garbage men, and they go around picking up whatever they find on the ground, right? And so if you have a cell that has popped open and all of its DNA is everywhere, some other bacteria or archaea is going to come around and pick it up and use it. All right, the second way that you can get genetic variation through gene transfer is transduction. So this is when a virus picks up DNA from one prokaryotic cell and transfer it to another cell.
Just like a mosquito.
It is a little like a mosquito, right? Mosquito will take a disease from one person to another. In this case, a virus is taking DNA from one cell and giving it to another cell. And then the third one is conjugation. And that is when genetic information is transferred directly by cell-to-cell contact. So the way this happens is you have 2 bacterial cells and they'll form a tube between them, and then they will transfer genetic material, either a plasmid. Do y'all remember what a plasmid is from 1113?
Yeah, it's like a little ring of genetic material. It's separate from the main DNA.
Yep. Or they could put part of their main DNA, right, and transfer that. So either of these things is possible. Okay, so bacteria look different from each other under a microscope. They are different in size, shape, and motility. So most bacteria are about 1 micrometer in diameter.
What's that weird letter?
Oh, that's a mu.
What?
It's a mu. So it's a micrometer.
Okay.
So yeah. And so this is a very, very tiny bacteria that we have found called Mycoplasma mycoides. It's very small. And we can compare that to the largest bacteria we know about, which is Thiomargarita namibiensis, which is responsible for swamp glow. And so you can compare the sizes.
I can't even see the little one.
It's that little green dot right there.
Okay. But you know I'm colorblind.
Yeah, I know. You're a dog. All right, the second thing is bacteria come in different shapes. So some of them come in rods, like a stick, right? And some of them come in spheres. And so the spheres will kind of stick together and make chains. And then some of them have a spiral shape. It could be a very tight spiral or a really loose spiral. Okay, so that's shape. And then the motility varies. So what's motility mean?
It means your ability to move.
Yeah. So some bacteria are non-motile, meaning they don't move, but they'll just swim or glide. And so these are some swimmers here. And then Oscillatoria, which is this beautiful green bacteria, doesn't really swim, but it can kind of drift around or glide a little bit. All right, so another way that people have used to tell the difference between different kinds of bacteria is to look at their cell wall. So a long time ago, people maybe didn't have as good microscopes as we have now, but they knew that if they took a particular stain called a gram stain, that some cells would turn purple, so they'd be gram positive, and other cells would stay pink. Those are gram negative. So you can see here, this is a mixed culture of 2 kinds of bacteria, gram positive and gram negative bacteria. We know now that the gram-positive cells look like this, and they have lots of this carbohydrate peptidoglycan. Okay, excuse me. And this will take up the purple dye and make these cells, purple when you dye them. Gram-negative cells will still look pink. That's just kind of the baseline color of the dye. And that is because the peptidic glycan is sandwiched between two polysaccharide layers, and so the dye can't really get through to it and make it a color, so these guys will stay pink. Okay? And the implication of this kind of matters in terms of functionality too, right? So we'll talk more about this later, but just keep in mind that the gram-negative ones have this extra kind of membrane on the outside. Okay, so what's an organic molecule?
Oh, I know. It's one of the ones with the carbon background and like hydrogens coming off of it.
Yep. You got one?
Methane.
All right. Anything else?
I can't think of anything.
Can't think of anything? How about sugar?
Yeah, sugar.
All right. How about starch?
Yeah, starch.
All right. Well, what's an inorganic molecule?
It doesn't have the carbon backbone. I can't think of any examples, though.
I can't think of any examples either because I only ever study organic molecules, but I'll let you think about that. All right, let's talk about metabolic diversity. So, no matter what kind of organism you are, you got a couple of things you need, right? You have to be able to get chemical energy into your body and use it to make ATP. So what do we do with ATP?
It's like the energy source that powers cells.
Yeah, so we need ATP to power our cells. Okay, but we also need carbon compounds that serve as the building blocks for the synthesis of cellular components, right? So all of our components are made out of organic molecules with that have all these carbon-carbon bonds in them, right? So we need to get those carbons from somewhere to be the building blocks of the molecules that we need, okay? So you need two things. You need ATP and you need carbon compounds. Make sense?
Yeah.
All right, so we have different ways you can make ATP. You could be a phototroph. So you know what photo means?
It means light.
Yeah. So phototrophs use light to excite electrons. And when they do that, they make their ATP through photophosphorylation, which you learned about in 1113, right? Do you remember what phosphorylate means?
Yeah, it means put a phosphate on it.
Yeah, it means put a phosphate on it, right? So we're using the light of energy to put a phosphate on what?
ADP.
Yeah, to make it into?
ATP.
All right, good job, Bird. All right, so you could also, instead of being a phototroph, be a chemoorganotroph. And so in this case, you're oxidizing high potential energy organic molecules, right? So you have an organic molecule. It's probably a large complex molecule with lots of bonds, and you're going to oxidize it. What does oxidize mean? Do you remember from 1113?
Yeah, it means like you're losing electrons, but you're also losing like hydrogens.
Yeah, to oxidize means you're losing electrons, but for the sake of what we're doing in biology class, usually you can tell you've lost electrons because you've lost a hydrogen as well, okay? All right, so you're basically you're losing hydrogens from high potential energy organic molecules. And so for that, you are making ATP by cellular respiration or fermentation. And then finally, so I should point out, so that would be through cellular respiration or for fermentation, you're starting out with a large molecule like sugar, for example, glucose. All right, chemolithotrophs are oxidizing high potential energy inorganic molecules, meaning they're not eating sugars, they're not eating fats, they're eating an inorganic molecule and using the potential energy locked up in that molecule to be able to make ATP. And the way that they do that is through cellular respiration. All right, so those are the three ways you can make ATP. Now you have two ways that you can make carbon compounds. You can be an autotroph, and that means that you're making these building block compounds from simple starting materials. So you're actually putting together the building blocks. Heterotrophs are absorbing building block compounds from their environment. So maybe they're eating them or absorbing them in some way. Okay. And so if you look at those 3 different ways to make ATP and 2 different ways to make carbon-carbon bonds, this gives you 6 different combinations of metabolic diversity that you could have. All right, so I have passed out all of these cards that I want you to hold up when we get to your correct turn, okay? All right, so if you get your ATP from sunlight, and you get your carbon-carbon bonds by synthesizing themselves, you're in a photoautotroph, okay? There you go. So you say your part.
Okay, phototrophs, they use the sun to make ATP. Autotrophs make their own compounds from carbon dioxide.
Okay, good. All right, let's move on to photoheterotrophs, okay? So photoheterotrophs, you want to say your part?
Yes, they use the sun to make ATP.
Right, and where are they getting their carbon-carbon bonds from?
They're absorbing compounds from other organisms.
Okay. All right, let's talk about chemoorganoautotrophs.
They use organic molecules to make their ATP, like, cellular respiration or something.
Yeah.
They are making their own compounds from carbon dioxide, etc., like I just said.
Okay, good. All right, what about chemoorganoheterotrophs?
They're making their organic molecules. They're using organic molecules to make ATP.
They're absorbing compounds from other organisms. That's where they're getting their carbon-carbon bonds from.
Okay, good. And then the last ones, chemolithoautotrophs.
I had to do a second one because there's not enough of us.
Well, that's fine. So what's a chemolithotroph do?
It uses inorganic molecules to make ATP.
Okay. And the autotroph?
Make their own compounds from carbon dioxide, etc.
What about a chemolithoheterotroph?
They're using inorganic molecules to make ATP. They're absorbing compounds from other organisms.
All right, so these are all the different compounds. So just pay attention to breaking down the word to figure it out. And just remember, every single one of these, they are getting their carbon from one place and they're getting their ATP from someplace. Just figure it out. Okay. So as you can see, there's a great deal of metabolic diversity in the prokaryotes, much more than you would expect to find in any other lineage. So the basic chemistry that was required for photosynthesis and cellular respiration and fermentation originated in these prokaryotic lineages. And because there are so many different variations on these metabolic processes, prokaryotes have been able to diversify into millions of species that occupy diverse habitats that can't be occupied by anything else. Okay. So I'm going to remind you now about how ATP is produced through cellular respiration. What I would ordinarily do if we were all together is I have this great interactive activity where we use ping pong balls to illustrate the electron transport chain. Okay, so I'm going to talk about that in a minute. Unfortunately, we can't do the ping pong balls, but all right, so let's talk about the fact that there are millions of bacterial, archaeal, and eukaryotic species that are chemoorganotrophs, okay? Meaning they break down organic compounds to get energy to make ATP. So it could be something like breaking down glucose, for example, okay? And the way this works is the enzymes are stripping electrons from the organic molecules and transferring them to electron carriers. So remember, we have a bunch of proteins that are embedded in the cell wall, right? And these proteins are each more electronegative than the one next to it, all right? And so the first one is a little bit more electronegative than the surrounding area, and it'll snatch an electron, okay? this protein here is more electronegative than that one, so it'll snatch that electron away from it. This one is more electronegative than the other one, it'll snatch that electron away. Okay. By the way, each time one of these proteins is snatching an electron away from the other member of the electron transport chain, the energy that is released is used to boost a molecule, a hydrogen proton across the membrane from inside of the cell to outside of the cell, right? So when it gets snatched away over here, it boosts this molecule, this proton outside. When the electron gets snatched away again, it boosts another hydrogen outside. Oh, look, this one's missing an arrow. When this protein snatches the electron from this protein, it boosts another molecule, another hydrogen proton across the membrane, etc. The end of the line for the electron transport chain could be many things, but when we learned about this in 1113, the example that we usually give is oxygen. So for aerobic organisms, oxygen is the final member of the electron transport chain, right? After all the electrons are snatched, one from the other and the other, and the electrons stick at the end with oxygen, you end up with a giant pile of these hydrogen protons all stuck outside this membrane, okay? So there's a lot of them here, a lot of hydrogen protons here, and not so many over here, okay? So that creates a gradient. So if you have a pile of something in one place and not so much in another place, just using a passive process like diffusion, like which way would these hydrogen protons want to go?
They want to go to the left, where it's less crowded.
Yeah, they want to go to the left, where it's less crowded, right? But the membrane is stopping them. However, there is a molecule called ATP synthase that is stuck in the membrane here, and it works like a water wheel, and basically it allows the hydrogen protons to go through it, and when they go through it, it turns it. And every time it turns, that energy is transferred into making a molecule of ATP. So the hydrogen protons rush past, the ATP synthase molecule turns and turns and turns, and it makes ATP. Okay, that is the electron transport chain. That is phosphorylation. Okay. And then what you can see here is that you're starting out with a very high potential energy donor over here, and the electron is transferred to the final acceptor, which is in aerobic organisms is oxygen, right? And that energy is harnessed to make ATP. Okay. In cellular respiration, like you learned about in 1113, most eukaryotes use organic compounds with a really high potential energy as the original electron donor, and then they use oxygen as the final electron acceptor. As you learned, when we undergo aerobic respiration, we make carbon dioxide and water as a byproduct. But with bacteria and archaea, they can live in places where we don't have these organic compounds as the energy donor, and we don't have oxygen as the final electron acceptor, okay? And so we can see here, so here's us, right? We're organotrophs. We start out with, let's see. Well, we start out with sugars as our electron donor, and we produce carbon dioxide as a byproduct, and then oxygen is our electron acceptor, which produces water as a byproduct, right? Okay, but there are organisms that live in places where there isn't oxygen, and so instead they'll use sulfur products or carbon dioxide or even iron, like we learned about, to be able to accept that final electron in the electron transport chain. There's also organisms that don't have access to organic compounds like sugars, and some of them will use other kinds of compounds as their electron donor. We learned about fermentation in 1113 too, right? So this is how you can make ATP without using electron transport chains. This is a process that's much less efficient than cellular respiration and doesn't use an outside electron acceptor, so you can do it without oxygen. And so you can have various organic compounds as a starting point. So Clostridium bacteria will ferment complex carbohydrates or proteins or purines or amino acids. And you have other bacteria that ferment lactose. So It's lactic acid fermentation. And you have bacteria in the digestive tract that ferment carbohydrates, and that's how we are able to digest the carbohydrates that we eat. All right, so like I said, we don't just need ATP, we also need carbon-carbon bonds. So heterotrophs have to consume these kind of building block compounds from other foods that they eat, right? But autotrophs are able to make their own building blocks. And so just some examples of autotrophs that make their own building blocks. There are organisms called methanotrophs, which are methane eaters, and they use methane as their carbon source. And there's other autotrophs that are known as methanogens, and so this is a kind of an archaea that produce methane as a byproduct of cellular respiration. So this is what is causing, for example, cattle produce a lot of methane. And people always think, oh, methane, it's being produced, by, I don't know, cow farts or whatever. But no, it's actually when they exhale, the methane from the archaea is coming out of their digestive tract and is just being exhaled with carbon dioxide. All right, so it's important to remember that there was no free molecular oxygen on the Earth for 2.3 billion years. So a world without any oxygen. We think that the first lineage of photosynthetic bacteria were the cyanobacteria, and they were the first to use water as an electron source for photosynthesis, and they were the ones that changed the atmosphere of the Earth by adding oxygen to it. All right, so why was oxygen necessary for aerobic cellular respiration?
It was the final electron acceptor.
It was a final electron acceptor. Before oxygen was present on land or in the ocean, cells could only carry out aerobic respiration. So before there was oxygen, they could only carry out anaerobic respiration, right? So aerobic means with oxygen, anaerobic means without oxygen. And so anaerobic respiration, it could be fermentation, but it could also be respiration that used something other than oxygen as the final electron acceptor. Oxygen is a very efficient electron acceptor. So you get more energy released in the electron transport chains that have oxygen as the ultimate acceptor than if you use other acceptor substances, right? So if you have access to oxygen, this is the drop in free energy relative to glucose. So here's your glucose molecule, has lots of potential energy, right? How much energy can you harness using different electron acceptors? Well, if you use oxygen, you can harness the most, okay? You can get almost 700 kilocalories per mole when you use oxygen as the final electron acceptor. There are bacteria, though, that can live life without oxygen. And so if they use nitrate or iron as a final electron acceptor, they can survive and produce ATP, but it's much less efficient process, right? So it's kind of harder to live in an area where iron or nitrate is your final electron acceptor. All right. So everything that is alive requires nitrogen to make proteins and nucleic acids because nitrogen is involved in those molecules. And nitrogen is abundant in the atmosphere, right? It's everywhere. But most organisms can't use it by directly just taking air from the atmosphere. They have to get nitrogen from either ammonia or nitrate, which is NO3-. And so there is a process called nitrogen fixation that certain bacteria and archaea can do that can convert N2 to NH3. Okay, so this is a process that takes atmospheric nitrogen and puts it into a form that, for example, plants can use. So we've referred to nitrogen-fixing bacteria as the bacteria that can take this atmospheric N2 and make it into NH3. And a lot of these kinds of nitrogen-fixing bacteria will live very closely with plants. Some of them induce the plant to make it a little nubbin in its roots to live in, and those are called root nodules. And so it's a very close association between the bacteria and the plants. So let's think about how nitrogen fixation fits into the nitrogen cycle of the Earth. All right, so we start out with nitrogen in the atmosphere. Like I said, you got them bacteria and archaea that can fix it, meaning take it from N2 and making it into another form like NH2, okay? So now you have an organic compound with an amino group on it after it has been fixed. And then that can get decomposed by bacteria, archaea, and fungi into ammonia, NH3, or ammonium ions. Okay, so after that, then you can have bacteria produce nitrite over here as a byproduct of respiration. And then this nitrate is used as an electron acceptor in other species. So, instead of having oxygen as the electron acceptor, nitrate can be used as the electron acceptor. This is then converted to molecular nitrate right here. And so that is a form in which it can be used directly by plants and algae, okay? And then later, the nitrate will get converted back into the atmospheric N2 by another set of bacterial and archaea species. And so this basically forms the nitrogen cycle. So it drives the movement of nitrogen atoms, through the ecosystems around the globe. So you have this part here where it's locked up in organic compounds, and then also taken from the atmosphere and released back into the atmosphere. We know that adding nitrogen to our crops helps them to be able to reproduce or to produce more biomass, right? But we also know that if you use too much NH3 fertilizers, it causes pollution. And so when you add NH3 to the soil, the bacteria will use it as food, and then the bacteria will then release nitrate or nitrate as waste products, and then those get washed out into the water and can cause serious problems in aquatic environments. And so what can happen is that the nitrates can decrease the oxygen content, and it causes dead zones to develop. And so you can see here that it stimulates blooms of things like marine algae and cyanobacteria. And then these will grow and grow and grow and grow until they use up all of the possible food, and then they will die. And when they die, it creates what's called an anoxic dead zone. So they've kind of blocking the oxygen, so no one can get at any of the oxygen, and the water's just filled with a bunch of dead stuff. All right, let's look at diversity in bacteria. So bacteria are a monophyletic group, so we know that there's at least 29 major lineages, phyla. And so we can tell them apart by, some of them by morphological characteristics, or by sequencing their DNA or their RNA and doing a phylogenetic analysis of the gene sequence data. All right, so the first lineage is the actinobacteria. And so this is very common in soil and freshwater habitats. Like when you breathe in and you smell this kind of earthy smell, like when it's, rain or whatever, that's caused by actinomycetes. So this is a particular kind of bacteria that gives soil its earthy smell. We also get streptomycin from actinobacteria as well. All right, the second lineage are the chlamydiae. So chlamydia is the disease, and it is basically a bacterial disease. It's sexually transmitted, and it has some serious consequences for women, it can lead to ectopic pregnancy and infertility. It can also cause a variety of other health problems, and in places that are, kind of developing and don't necessarily have as good medical care, people will frequently suffer from blindness due to chlamydia. Third group is cyanobacteria, so these are photosynthetic, so you can see they're green, okay? And we think this is the group that is responsible for the origin of the oxygen in our atmosphere, so you can go thank a cyanobacteria for that. Next, we have the firmicules, and so this is a group that's found in the human gut, and so they're usually in either rods or spheres. Some of the notable members of this group are botulism. Botulism is a disease that's created by a particular bacteria, just secreting a toxin into its environment. And Bt is another one. So Bacillus thuringiensis makes Bt toxin, which is a very important insecticide. So it's what do you call it? It's sprayed over crops, and it creates this this toxin that puts little holes in the intestines of like the caterpillars that eat it. There's also like BT crops that are plants that grow their own BT instead of just having it sprayed on the top. Proteobacteria. So these, it's a pretty diverse group, but they include like E. coli, which is the workhorse of most molecular biology, and members of this group do all sorts of things that we don't like them to do, as well as being, kind of good lab animals, or sorry, not animals, good lab creatures. They can also cause cholera, food poisoning, plague, dysentery, and typhus. And then finally, the spirochetes, which are named after their spiral shape. I don't know if you can see it here. They're not zigzag, they're like actual spirals. And they are found in diseases like syphilis and also Lyme disease. All right, so archaea are found nearly everywhere, including extreme places. And so there's several major phyla. And so there's the one phylum that's involved in these kind of extreme environments, like very hot, high pressure, acidic environments. They also include like the methanogens, and and the third group, which is found in saltwater and freshwater habitats. In terms of what you need to know about the bacteria and archaea, I am not expecting that you know the names of the key lineages, let me go back, of the bacteria or the archaea, okay? But you do need to know, if I say chlamydia, you need to know it's bacteria, okay? If I talk about, cyanobacteria, it's kind of in the name. But, if I talk about syphilis or Lyme disease, you need to know that it has a bacterial kind of, that it's a bacterially based disease, okay? You don't need to tell me that it's a spirochete. Does that make sense? So you don't necessarily need to know the name of each of these lineages, but you need to know that all of these things are bacteria, and you need to know that all of these examples of things that I gave you are archaea. By the way, there's a couple of other groups of archaea. I just put them here in case you're like a super archaea fan and you're like, hey, you didn't talk about the other two groups. But there they are. Okay, so hopefully I talked about prokaryotes, and you saw how they were different than eukaryotes, and we talked about how and why we study them, and we talked about 4 major themes and how bacteria and archaea have diversified, including gene transfer, metabolism, morphology, and ecological diversity, and then we just kind of went through a list of the key lineages of each group.